my stitches
Okay,
taken out
let's
Wednesday.
start. I thought maybe what we could do today is begin by reviewing all of cellular respiration. We'll then finish up cellular respiration, which is chapter 7. So we have just a little bit left to talk about. That's fermentation reactions and how it is that we handle other molecules within cellular respiration that aren't sugars. And then we'll start mitosis. Hopefully mitosis will be mostly a review for you guys from your high school biology class. Okay, so cellular respiration begins outside the mitochondrion, right? So this reaction, glycolysis, occurs entirely within the cytosol. And remember, the purpose of glycolysis is just to take a glucose molecule and to break it down into pyruvate. So it's the partial digestion of sugar to create a pyruvate molecule. And in the process of doing that, we also create some other things, right? We create a little bit of ATP and we create a couple of electron carriers. But the reason that we want to make pyruvate is because we want to get pyruvate into the mitochondrion, and that's the format in which sugar can be transferred across that membrane. So the way that we do that is first we destabilize our glucose molecule, and I suppose I ought to label that as glucose. So how is it that we destabilize glucose? Yeah, we ingest a couple of ATPs, right? So we take one ATP and we drop off its terminal phosphate onto our glucose molecule. And that makes ADP, right? So we go from adenosine triphosphate to adenosine diphosphate. And we do that twice. So why is that unstable now? Yeah, it's got two large negatively charged molecules on it, right? And phosphates are inherently destabilizing. Okay, so we take these phosphates and we stick them onto our glucose molecule, and that makes it unstable. And then ultimately it's going to break, right? And when it breaks, one of the phosphates from that original reaction is going to end up on the top of that one, and the other one's going to end up on the bottom of this 3-carbon chain. Now we've broken a bond, which means that what was released? Energy in the form of, we're going to make an NADH, and NAD picks up what? An electron, right? So we break a bond, and we liberate an electron. We pick that electron up with NAD+, and that becomes NADH. And that happens actually twice, right? So we're going to create 2 NADHs at this stage. Okay. That's not the only thing that happened here. What else happened at this stage in glycolysis? Yeah, keep going. Perfect. So we get 2 inorganic phosphates, right? And so we call them inorganic phosphates because they're not attached to any molecule. They're just floating around in the cytosol. And we get 2 of those inorganic phosphates that join the reaction and end up on the bottom of this one and the top of this one. So each of those 2 3-carbon chains has 2 phosphates associated with it. Okay, so this stage right here that's a redox reaction, right? What is a redox reaction? Oxidation and reduction, exactly right. So something gets reduced and something gets oxidized. What's being oxidized? Close.
NAD+.
these 2-3 carbon chains, right, when they're breaking, they're losing electrons, and they lose electrons, making it an oxidation reaction. NAD plus gets reduced because it picks up those electrons. Now, our electron carriers are important. And why are they important? Where are we going to be using our electron carriers in all of cellular respiration? Yeah, all the way down here at the electron transport chain. So all of the electron carriers that we're making ultimately are going to be shuffled down to the end and used in the electron transport chain. Okay, we have the phosphorylation event occurring right here. Is that substrate level or is that oxidative phosphorylation? It's oxidative phosphorylation. And what's the difference between oxidative and substrate level phosphorylation?
Substrates to make ATP.
Okay, so in one of them, a phosphate is transferred from a substrate or another molecule onto something else, right? And that's not happening here, right? Oxidative phosphorylation is where we're taking a phosphate out of the cytosol and sticking it onto something else. So this is oxidative phosphorylation of these two molecules. Okay, now...
Can you repeat that?
Yeah, you bet.
Taking
So the difference between substrate level and oxidative phosphorylation, substrate level phosphorylation, that's where you're taking a phosphate from one molecule and putting it on another. And then with oxidative phosphorylation, you're taking that phosphate out of the cytosol and putting it on a molecule. So it's just dissolved in the cytosol, or you put it on the molecule. Okay, so these next two stages are the same, right? And what happens at both of those next two stages? We're going to create something. What do you create? ATP, exactly right. So we take an ADP, or an adenosine diphosphate, an ATP molecule with just 2 phosphates on it so far. And we transfer one of the phosphates from one of these molecules onto that ADP to make ATP. And we do that twice.
But I don't know. But the last one where it's like green, I don't know why, but in my notes I put like on top where the NAD plus, I put reduced.
Yeah, NAD plus is reduced.
Oh, okay.
Yep.
Just making sure.
This molecule that becomes this molecule is oxidized,
Okay.
and this one is reduced.
Okay.
Yep, your notes are correct. Okay, substrate level or oxidative phosphorylation?
Substrate level.
It's close to oxidative, it's substrate level, right? Because we're transferring from this molecule onto that molecule, so that's got to be substrate level phosphorylation. And that happens in this reaction right here. We take 2 more ADPs, and we transfer those 2 remaining phosphates on to make 2 more ATPs. So the pyruvate is an unphosphorylated 3-carbon chain.
Okay, so the first, again, so the green one, it's the oxidative phosphorylation, and then the second one is the substrate level.
This phosphorylation event right here is oxidative.
Okay, okay.
And this phosphorylation event right here and right here, those are substrate level.
Okay.
Yep. Okay, so then what we're going to do, this is the end of glycolysis, right? That's the entire reaction of glycolysis. And when we're talking about fermentation in a few minutes, we're going to be dealing with just this reaction. Nothing else happens downstream from that. But in the presence of oxygen, what we can do is we can take pyruvate and we can move it inside our mitochondria. So this is the mitochondrion. We have an outer membrane and an inner membrane. And so this first step of the Krebs cycle occurs in that intermembrane space. What's happening here at this first stage? Do you recall?
And we're breaking apart the 3-carbon chain
Okay.
by taking one of them and making CO2, and then NAD plus is transformed into NAD.
Exactly. And how do we get... So, Sydney, what you're saying is we make a carbon dioxide, and then we also make an NADH. So what that means is NADH was reduced, but where did it get that electron for the reduction reaction?
They're floating outside.
What's that?
Aren't they like floating outside and then they come in with the Ubers? Is it that one?
Are you talking about this molecule right here? Yeah, that's eventually going to happen, Sophia. But
I
we're
just
gonna,
remember
we're
you
gonna,
calling it Uber, so I'm like, all right.
yeah.
Okay.
And that's what these are. They're Ubers. They're just transferring electrons about.
Okay.
And ultimately, we're going to need to get like these ones all the way in here, transferred into the mitochondrion. But we'll come back and talk about that in just a few minutes. So where did this electron come from to reduce NAD+?
3 carbon bonds, so energy was released and.
Yeah, and what's that 3-carbon molecule called? Pyruvate. Yeah. So pyruvate was broken. One of its carbons was taken off, and when we broke that bond between those 2 carbons, this one and that one, an electron was released, and that was picked up by NAD+. So this too is a redox reaction. What was reduced?
Pyruvate.
Close to pyruvate. What was reduced?
The NAD+.
NAD+ was reduced, right? Because it picked up the electron. You guys are right in that pyruvate was oxidized because it lost an electron. Okay, so we attach that 2-carbon chain to this thing, and that's called coenzyme A. And so this entire complex right here we call that acetyl CoA. And this whole reaction was catalyzed by an enzyme. And what was that enzyme called? Pyruvate dehydrogenase, exactly right. Okay, so coenzyme A is not more than a carrier of that remaining pyruvate molecule, those 2 carbons, into the Krebs cycle proper. So what happens, right, is coenzyme A brings these 2 carbons over here and drops them off on this molecule, which is called oxaloacetate. So oxaloacetate picks up those 2 carbons, one ends up here and one ends up there, and then we're able to move those into the Krebs cycle. What happens to coenzyme A? Yeah. Exactly right. So CoA is able to return back over there. And it just ends up being a shuttle, right? It picks up pyruvate carbons, takes it to the Krebs cycle, and then returns. It picks up more pyruvate carbons and then returns. Okay. So we're right here, and we have this intermediate, and now we need to start taking carbons off, right? So what's occurring right here, in between here and here? What happens in that state? A bond is broken, right? We take that top carbon off, and what happens to that top carbon? It becomes CO2. And because we've broken a bond, we're able to make a what? Yeah, we're able to reduce an NAD plus into NADH. What kind of a reaction is this? It is an oxidation and a reduction. So collectively that's called a redox reaction. Right? Because this molecule, whatever the heck that is oxidized. A carbon is broken off of it and an electron is released. That's oxidation. And then NAD plus picks up that electron, so it becomes reduced. All right, so what happens right here? Between here and here?
It happens again.
Same thing. Right? Because we're going from a 5-carbon chain to a 4-carbon chain, which means that one of those carbons needs to be lost as carbon dioxide. And because that carbon is broken off, it becomes carbon dioxide, we're able to liberate an electron and pick that up with NAD+. So that too is a redox reaction. Now this next stage right here, where we start shuffling carbon molecules and side groups around, this was the really weird one, right? And what ultimately do we want to make between here and here? We ultimately want to make ATP. And how do we do that? Yeah, we use GDP. So we have this little molecule that's very much like ATP, just with a different nitrogenous base on it. And GDP is able to pick up a phosphate to become GTP, right? And was that, what kind of phosphorylation event was that? So it's close to substrate level. Yeah, at this stage, right, where GDP is actually being phosphorylated, this is the oxidative stage. So we have a phosphate that's just kind of floating around inside the cytosol that gets stuck onto GDP. So that's oxidative. But we know that GTP is very short-lived, right? It's just really a transfer molecule. So it's going to drop its phosphate off on something else, and that something else is ADP, right? So ADP picks up that phosphate from GTP. And so for those of you who said substrate level phosphorylation, you're exactly right here, because that phosphate is transferred from GTP to ADP. Yep, Mac.
Sorry, what does PI?
So that's inorganic phosphate. That means that
Okay.
it's just floating around inside
Okay.
the cytosol.
Thank you.
You're welcome. Okay. You okay with that idea?
Yes.
Okay. So then we do some more molecular reshuffling right here, and what do we make? Yeah, so we make two more electron carriers, right? We make another NAD plus to NADH, and then we also make an FAD plus to an FADH2.
And both of those are electron carriers.
They're both electron carriers, and they both do exactly the same thing. However, they don't produce the same amount of ATP.